All right. Hello, everybody. Very good. Okay, very good. All right. In the name of God. So, this class is going to be about immune response against infectious agents, what we call infectious disease immunology. And it's one of the very crucial parts of immunology. It's basically a subfield of what is called infectious disease medicine. And infectious disease medicine is very important to uh, all physicians and all people who are involved in biomedical sciences, directly or indirectly, for historical and practical reasons. For historical reasons, because infectious diseases have been with the human beings all throughout history. If you look at history, you will see many examples of epidemics or pandemics that have influenced human societies or even human history. One of the examples that I'm showing a painting of in here is the Black Death epidemic in Europe in 14th century. So plague, which is an infectious disease caused by a bacterium named Yersinia pestis, is a good example of how infections and infectious diseases can influence human societies, how they can shape history, and how they can lead to radical changes in human societies. Black Death epidemic was caused by a form of plague which is named bubonic plague, and it has a story behind it. So if you look at this map in here, this is a map of the Mongols' invasion, Genghis Khan and his basically offspring from Mongolia in uh, basically 13th century. 13th century. So they started their invasion from this region, from Mongolia, and they invaded the first basically Sutherlands, China, and then they moved westward towards the Persian Empire and Abbasid Caliphate at the time. So they came, one group of the Mongols, which were basically headed by Genghis Khan himself. They attacked China first, and then they moved westward towards the Iranian Empire at the time. They sacked Bukhara first, and then Nishabur in 1221, and then the Genghis Khan's offspring, Hulagu Khan, was basically the next who launched a major invasion, and he went all the way to Baghdad. You probably heard about the siege of Baghdad in 1258 and the destruction of the House of Wisdom, or Bayt al-Hikmah, which was one of the major cultural and scientific centers at the time. So these are a group of Mongols that basically invaded lands south of the Caspian Sea. Another group of Mongols invaded lands north of the Caspian Sea, and they were naming themselves Golden Horde. And they went all the way to this region, which is a peninsula that you hear about it a lot these days, these few years actually. It's named Crimea, and it's a disputed region between Russia and Ukraine. So Mongols reached a city in Crimea, which was named Kaffa at the time. I think it's named Theodosia now, and they led siege to Kaffa. So this is the story behind the epidemic of bubonic plague. So this was years like 100, almost 100 years after the initial invasion by Genghis Khan. So this is 1344, 1345, that time almost. So they laid siege to Kaffa, but they could not penetrate the city because it was protected by big towers and walls. So they were using catapults to throw stuff inside, but they could not penetrate the walls. So the siege lasted for almost a year until something very bad happened to the Mongol army. So one day, soldiers and officers went to the commander of the Mongol army and they told him that some of our soldiers are dying from plague. So plague was very well known to Mongols because before the occurrence of plague in Europe in 14th century, East Asia, including Mongolia, had experienced limited outbreaks of plague. So they were very familiar with the disease, and they knew that this is a contagious disease, especially when there are a lot of people gathering in one region. It's very likely for the plague to be spread. And the way that plague spreads, well, we have a form of plague which is transferred between humans through respiratory means. It's called pneumonic plague. But the plague that was more common at the time was not being spread between humans. It could be spread between in populations by flea bites, FLEA, flea bites. So Yersinia pestis has an animal reservoir like mice and rats, especially rodents. So fleas bite rodents and then they bite human beings. And through this biting, they transfer the disease. So it's not transmitted between humans, at least the plague that was common at the time. It's transmitted from rodents to humans. Nonetheless, it's a contagious disease. So the commander of the Mongol army ordered the army to retreat as fast as possible because he knew that if they leave the army, they continue the siege and they leave the army in a basically localized region, probably all of the soldiers are going to die. So that was a wise act. But before leaving the city of Kaffa, they did something very nasty, I can say. So they said that, well, this plague that has affected us, it has made us the basically leave the city alone. But what we can do is to inflict the same thing on people inside the city. So they put the corpses of dead people who had died from plague, into the catapults and threw those corpses inside the city. We don't know exactly how, maybe hundreds, maybe a few hundred corpses of dead soldiers were thrown into the city of Kaffa, and then they left afterwards. So plague spread in the city of Kaffa. So probably, well, those corpses, their clothes were contaminated with fleas that were carrying the disease. We don't know exactly how, but the disease spread in the city of Kaffa, and it was a peninsula. So, and it was a port city. So people inside the city started moving all through the Europe. A lot of them were Italian merchants who used maritime routes to go back to Italy, and the disease was spread all throughout Europe, and that was the beginning of the bubonic plague. This act of throwing the corpses of dead people inside the city during a war, it's some people call it as the first documented example of biological warfare. You know, biological warfare that you hear about it in different news media or different resources. It's something that is relatively a modern concept after the scientists discovered that microorganisms exist, and some of these microorganisms, like the one that causes anthrax, for example, can be spread easily. So some people used it for bioterrorist attacks. But this is an example of bioterrorist attack, a biological warfare, a bioterrorist attack that happened almost like 700 years ago, the siege of Kaffa, and it led to bubonic plague epidemic, and it's killed almost a third of the European population at the time. So it has a story behind it. This is another painting that shows or highlights another historical event which was closely associated with an infectious disease. The guy in the middle is Napoleon, and it's his army. And as you can see from this painting, the army is in disarray. They don't look to be in a good condition. So this is the story of Napoleon's invasion of Russia in 1812. So they went and captured Moscow, but Russians, before retreating, they had burned all the farmlands and all of the supplies that the invading army could use. So they went to Moscow. Then it was winter, and they had problem with their supply lines, so they had to retreat. And the army was very big army. It was called Grand Army. So this is how it's written in French. And it was 500,000 professional soldiers, 500,000 soldiers, the biggest organized army that Europe had ever seen by that time. So it's said in historical records that when Grand Army returned to France, entered France, only 45,000 of that 500,000 soldiers were still alive. Like 90% of them had died. Some of them had died because of the lack of food and famine, and some of them had died because of a disease that was named typhus. Typhus is a bacterial disease that's similar to plague, can spread around when you have huge collections of people in close contact, and similar to bubonic plague that was transmitted by an insect, by fleas, FLEAs, fleas. Typhus can also be transmitted by lice, LICE, by lice. And it can be transmitted from one person to another person. So it doesn't need an animal reservoir. And it was called Army's Fever at the time, Army's Fever. And it was a nightmare for army commanders because when they were gathering their armies, especially leading siege to a particular region or city, typhus was one of the things that were threatening their health and their survival. This is another painting closer to the present time. It's from a journal that was being published in Paris, 1901, and it shows the basically widespread mortality caused by cholera. And this is the picture of a girl, one of the latest cases of smallpox from 1974. And unlike the previous diseases, unlike bubonic plague, typhus, and cholera that were caused by bacteria, smallpox is a viral disease. And it's closely associated with the history of immunology. You probably recall from previous classes that the first effective vaccine, was the one that was generated by Edward Jenner for smallpox. So in this class, we want to look at different types of immune response against infectious agents. And it's very important for us to know about them because, well, knowing the type of immune response would help us devise strategies to prevent the disease or even treat the disease. So there are three factors which are important in shaping the immune response against infectious agents. The first one is the type of the microbe or the pathogen, whether it's a virus, a bacterium, a fungus, or a worm. So that is important in determining the type of the immune response. The second one is the organ or organs that are infected. Is it a systemic infection that influences multiple organs, or it's an organ-specific disease? But the third factor, which is kind of more important, is the localization of the pathogen at the cellular level. Are we talking about a pathogen which lives inside cells, or are we talking about a pathogen which lives outside cells? So, is it an intracellular infection or an extracellular infection? So, the way that I want to organize immune responses is to bring the type of microbe in here. We have bacteria, viruses, fungi, and parasites. And by parasites, I mean eukaryotic organisms that can cause disease, like worms or some unicellular organisms. Put these four types of uh, pathogens on the left hand side of the page, and then two localizations, extracellular and intracellular, in here, and make a table to see what we have in here. So the majority of bacteria are extracellular bacteria, like E. coli that causes diarrhea, or pneumococci that can lead to lung infection or to meningitis. But we do have some intracellular bacteria as well, like Mycobacterium tuberculosis and Mycobacterium leprae, which are causative agents for tuberculosis and leprosy. These are intracellular bacteria. For viruses, well, viruses, in order to survive and in order to proliferate, they have to be inside cells. So these are intracellular pathogens. For fungi, the majority of fungi are extracellular, but we do have some fungi like histoplasma which live inside cells. And so is the case for parasites. Helminths, they're worms, they're too big to fit inside the cell, but we have parasites like Plasmodium, which are intracellular. And basically, it's a causative agent for malaria, if you recall. So I want to use this table to categorize immune responses. But before doing that, we need to refine this table a little bit, because intracellular comprises two subspaces, and extracellular also includes two subspaces. So each of these parts should be divided to two parts. And in order to show you that, for intracellular, imagine that I have a mycobacterium and a virus in this figure. So here on the left-hand side, I have some mycobacteria, and this is a macrophage. And on the right-hand side, I have some viruses, and here is a cell. So imagine that these are Mycobacterium tuberculosis. And the way that Mycobacterium tuberculosis enters our lungs is through inhalation. So we inhale the mycobacterium. So it goes inside the lung, it gets phagocytosed. They get phagocytosed by alveolar macrophages inside the lung, and they get circumscribed in something which is named the phagosome. So a lot of pathogens, when they get phagocytosed, they die inside phagosomes, because phagosomes will merge with lysosomes, and lysosomes have toxic stuff in them that kills the pathogen. But in the case of mycobacteria are fine with that. They can live inside the phagosome or inside the phagolysosome. They proliferate, they kill the macrophage, they come out, they get phagocytosed by another macrophage, and the cycle is repeated until we have damage to an area of the lung, and basically something is generated which is called mycobacterial tubercles, and hence the name tuberculosis. So Mycobacterium tuberculosis is an example of an intracellular pathogen. It lives inside cells. On the right-hand side, I have some viruses. Imagine that these are like hepatitis virus that infect liver. And here I have a hepatocyte, a liver cell. So viruses would infect the hepatocyte. They will go inside, they will start proliferating, they kill the hepatocyte, they come out, and they get internalized, or they infect another hepatocyte, and the cycle is repeated until the liver damage happens. So this is another example of an intracellular pathogen. The pathogen in the left side is an intracellular pathogen, but it's very different from the one on the right side. The one on the left side is circumscribed inside a vesicle. For that reason, it's called an intravesicular pathogen or intraphagosomal pathogen. But the one on the right side, it's floating in the cytoplasm, so it's called intracytosolic pathogen. So both of these are examples of intracellular pathogens, but this one is intravesicular and this one is intracytosolic. So, and it's very important for immune system to see it inside the vesicle, to see the pathogen inside the vesicle or inside the cytoplasm. So I have to subdivide it, I have to divide the intracellular space into intravesicular and intracytosolic. So is the case for extracellular space. When we say extracellular, sometimes the pathogen is at the surface of the epithelium without penetrating the epithelium, like at the surface of the skin or at the surface of the gastrointestinal epithelium. So this is one extracellular space. But sometimes the pathogen or pathogens penetrate the epithelium and they enter interstitial fluid or they enter blood or lymph. This is still extracellular. When I say blood, I mean blood fluid, the fluid part of the blood, not the cells. So we have to divide the extracellular to these two subspaces, epithelial and the interstitial blood lymph. And now if I look at this table, we have four groups of pathogens and two spaces, each of them divided to two subspaces, and we have 16 regions in here. So let's populate it. So here we have mycobacteria. Mycobacteria are intracellular, intravesicular. But we do have pathogens like rickettsia, which is the causative agent for typhus, like Napoleon's army's nightmare, which is intracytosolic, or another pathogen named chlamydia. For viruses, they're usually in the cytoplasm. For fungi, if they're inside cells, they're usually circumscribed inside vesicles. There are limited cases of fungi which might be intracytoplasmic. For parasites which are intracellular, leishmania is an example of an intravesicular pathogen. So it's inside cells, but inside phagosomes. But plasmodium, which is the causative agent for malaria, is intracytoplasmic. For extracellular, we have many examples. Sometimes an E. coli could be at the surface of epithelium, but sometimes it can go deep and enter interstitial tissue. A streptococci, sometimes they're at the surface of epithelium, but sometimes they can cause deep infections. And so is the case for some of the other pathogens. For fungi, when they are extracellular, they are usually at the surface of epithelium. There are limited cases of fungi which live inside blood or interstitial space. And finally, for extracellular parasites, a lot of worms, they live at the surface of epithelium, but we do have some worms that live in the interstitial space. So this is a table that shows a general overview of different spaces in our body at the level of the cells, intracellular, extracellular, not anatomical spaces, and different pathogens in these different spaces. So why this table is important? Well, because we can use it to categorize immune responses. So immune system has an innate part and an adaptive part. The innate part, well, it's physical chemical barriers as well as phagocytes. And the adaptive part is B lymphocytes and T lymphocytes. So if I'm a phagocyte, like a neutrophil or macrophage, it doesn't really matter for me where the pathogen is located, except for the epithelial surface that I don't have access to. The rest of these spaces I can access. If something is floating in blood or interstitial fluid, I can perform phagocytosis. If it's intravesicular, well, it's inside my own vesicles, because things that live inside vesicles are basically, these vesicles belong to the phagocytes. And if it's intracytoplasmic, well, I can phagocytose the entire cell that contains something inside it. Or I can wait for the cell to die, and then I phagocytose cell debris as well as the pathogen that has been released. So for phagocytes, it's not really different except for the epithelial surface to which they don't have access. It doesn't really matter where the pathogen is located. But for B lymphocytes and T lymphocytes, it's very important. So if I want to use this line in the middle to sort of divide the responsibilities between B cells and T cells, and I'm trying to do a modeling. A model is a simplified representation. I'm trying to make it simple, the simplified expression or simplified explanation. So if I want to use this line in the middle to separate responsibilities, if I want to give one of these two regions to B cells to defend, and one of these regions to, say, T cells to defend, which one should I give to B cells? B cells produce antibodies. Shall I give the extracellular space to B cells to defend? Or shall I give the intracellular space to B cells to defend? So you can communicate here, uh, just type extra intra. So the question is, should I give the extracellular space to B cells or intracellular space to B cells? If your answer is extracellular, type E or X. If your answer is intracellular to B cells type I or IN, that would be sufficient. Let's see. Okay, good. Yes, B cells produce antibodies and they have to perform the defense using antibodies. Okay, so the space that B cells can defend more efficiently is extracellular space. Why? Because B cells produce antibodies and antibodies cannot go inside the cells. Antibodies cannot penetrate the cell membrane, so they cannot go inside the cells. If something is hiding inside the cells, antibodies normally would not have access to that region. So antibodies would defend the extracellular space, and then we'll have to give the intracellular space to T cells. Later, we will see that this is an oversimplification because B cells and T cells would always work together. But as I mentioned, this is a simplified representation. So now I have a question for you. Some of you have injected, say, hepatitis B vaccine or vaccine for COVID-19, coronavirus. So viruses, as I mentioned, they live inside the cytoplasm. They live inside the cytoplasm. Nonetheless, when we perform vaccination like hepatitis B vaccination, in order to find out whether the vaccine has been effective or not, we go to a laboratory and they perform an antibody test. They perform an antibody test. So I got my Hep B shots. I got my hepatitis B vaccination. So it's a 3-time vaccination, 3-dose vaccination. And then after the third time, after a couple of months, I want to get myself tested to see whether the vaccine has been effective. So the test that I do in the laboratory is an antibody test. If I have developed sufficient levels of antibodies against hepatitis B, it means that the vaccine has been effective and I'm protected. If I have not developed sufficient levels of antibodies against hepatitis B virus, it means that the vaccine has not been effective. So, considering that viruses are all inside cells, this seems a bit paradoxical. So, if viruses are inside cells and antibodies cannot penetrate cells, how is it that when we vaccinate ourselves for viral pathogens, we expect our B lymphocytes to produce antibodies? So this seems a bit paradoxical. So the answer to this question is this. The table that you're seeing in here shows the final destination of the pathogen, like the final destination of viruses in the intracytoplasmic space, or the final destination of Plasmodium or Rickettsia chlamydia or intracytoplasmic or intracytosolic space. But the point is that for a pathogen to reach its final destination, it needs to pass through other spaces. Like if a virus wants to go inside the hepatocyte, it cannot go jump into the hepatocyte directly. So it has to go through other spaces. Like in case of hepatitis A, which is a fecal oral infection, it enters through contaminated water or food. At the beginning, the virus is at the surface of the intestinal epithelium. Then it penetrates the epithelium and enters the blood. Then it circulates in the blood until it reaches liver, and then it gets internalized by hepatocytes. So it has to be at the surface of epithelium first, then inside blood, and finally it will be intracytoplasmic. In the case of hepatitis B, well, hepatitis B or hepatitis C could be transmitted sexually transmitted disease. So if they are transmitted as an STD or a sexually transmitted disease, in the beginning, the virus is at the surface of genitourinary epithelium. Then it penetrates epithelium, enters interstitial space, and from there it enters lymph or blood, and then it circulates until it gets internalized by hepatocytes. If hepatitis B or C are transmitted through blood transfusion or maybe through needle sticks, in the beginning they are present in the blood or interstitial space, and then they will circulate until they reach the liver. So different pathogens, they have a point of entry, a destination, and a path in between. The beginning, the end, and the path in between. And I have made something like this in order to show different paths by pathogens. So in this, basically these curved arrows that I'm showing you in here, like the beginning of the arrow, I meant the point of entry. The end of the arrow is the destination, and the distance in between is the path that they take to reach destination from their point of origin. So some pathogens are like this. They establish themselves at the surface of epithelium. It's their point of entry. They remain at the surface of epithelium. They proliferate at the surface of epithelium. They cause disease there. So they do not penetrate any tissues. Like, for instance, the E. coli that causes diarrhea by generating toxins. It lives at the surface of epithelium. A lot of fungal infections of the skin, they always live at the surface of epithelium. They do not penetrate to deep tissues. But sometimes the point of entry is the surface of epithelium, but then they penetrate and go inside. Like the same E. coli can penetrate the tissues and go deep. So in the beginning, it's at the surface of epithelium, but later it penetrates and establishes the infection in the interstitial space. Sometimes they are at the surface of epithelium, then they enter interstitial space, they get phagocytosed and live inside vesicles. Sometimes like viruses, which are fecal oral viruses or sexually transmitted viruses, they are at the surface of epithelium first, then they penetrate, go inside interstitial space or blood, and then finally they reach their target cell and they get internalized. And we have other examples as well, like the ones that are starting from here. They do not need to traverse epithelium because they are injected by insect bites, for instance, or maybe by transfusion or needle sticks, and then they end up in different tissues or different spaces. So what we see from this very simplified representation of pathogenic point of origin, path, and destination is that all pathogens start their journey from extracellular space. This is important. Even the pathogens that want to reach inside the cytoplasm or the vesicles, they have to start their journey from extracellular space. And this means that this is the realm of the antibodies. The extracellular space is the kingdom of antibodies. The antibodies are present there and they always have a chance to protect. So that's very important to keep in mind. Before the pathogens become intracellular, they're outside of the cells, and antibodies can protect. Once they are inside cells, antibodies will not be effective anymore. And then we have to find other ways to protect. All right. So what I'm going to do now is to start from the intravesicular space, the one in here, and show you the types of immune responses that we have. Then we will be talking about intracytoplasmic space, and then we will be talking about extracellular space. So for intravesicular, the pathogens which are in here can be divided to two types. One group of the pathogens are the ones that get inside vesicles as a part of their life cycle. So they want to be there, they like it there. That's where they proliferate, like mycobacteria. We also have another group of pathogens which normally don't live inside vesicles, but out of bad luck, they get phagocytosed. So these are usually extracellular pathogens, but sometimes they get phagocytosed and they die inside vesicles. So let's see what happens for them. Immune defense against microbes in the intracellular space. So I have a pathogen in here. It gets phagocytosed. And once it's phagocytosed, lysosomes, which I'm showing with these red circles in here, will merge with the phagosome. And inside lysosomes, there are a lot of things that are detrimental for the pathogen. Like they have a low pH, acidic pH, which is harmful for a lot of microbes. They have an enzyme named lysozyme, which can lead to the destruction of bacterial cell wall. They have proteases, which digest microbial proteins, and some other molecules. And this is a very efficient way of killing microbes. It's a first line defense, and it's an innate type of defense. You know, innate and adaptive, this is an innate type of defense. And we also have another name for it, oxygen independent killing. It implies that we have killing mechanisms which require oxygen, but not here, not here. So oxygen independent killing. So this is the first step of defense against pathogens by phagolysosome formation. And as you can guess, the pathogens that are killed in here are the ones that do not live inside vesicles. They've been phagocytosed as a process, as a part of defense. So they don't like to be there, but they get phagocytosed. What about pathogens which can live inside phagosomes? Like Mycobacterium tuberculosis is a good example. So these pathogens have developed ways to survive inside phagosomes. So mere phagolysosome formation and oxygen-independent killing are not going to be sufficient for the removal and the killing of the pathogen. So here something interesting happens. Some of these pathogens might be, well, a lot of the pathogens that have been phagocytosed live there, but some of them might die, and some of them might lose some of their antigens. So some antigens derived from the pathogen are floating around in the phagolysosome. Then we have another vesicle inside the cell, which has something named an MHC molecule. It will go and bind to the phagolysosome. Antigens in the phagolysosome will get loaded at the surface of the MHC, and then the MHC will move to the cell surface and start showing the antigen to other cells. So what other cells? Well, we have two groups of T lymphocytes, CD4 T lymphocytes and CD8 T lymphocytes. If you have these groups of T cells, the ones that have the CD4 molecule at their surface will go and bind to MHC class 2, which is now showing the antigen at its surface. And it's the first step for the activation of T cell. From now on, this macrophage or dendritic cell will be named antigen presenting cell, APC, because it's presenting antigen to T lymphocytes. What happens then next is very important. In addition to the MHC and the antigen, another molecule appears at the surface of the T cell. And this molecule, which I'm showing with the red triangle in here, it's called the co-stimulatory molecule. You know, T cells are like very sleepy people. So some people, when they're asleep, if you call them or tap them, they wake up easily. But some people don't wake up that easily. So you call their name, but they like roll around a little bit, but they don't wake up. Then you have to use a stronger stimulus to wake them up. So T cells are like those sleepy people that don't wake up easily. So you have to give them MHC with the antigen as well as a second molecule, which I'm showing in here with the red triangle, and the name that I have for it in here is B7. So it binds to a receptor at the surface of the T cell, which is named CD28, and then this fully wakes up the T cell. How does it wake up? Well, it starts producing a cytokine named interleukin 12, interleukin 2, sorry, interleukin 2. And interleukin 2 will tell the T cell to start proliferating. It enters mitosis, mitotic division. So a lot of T cells will be generated, and all of those T cells will have exactly the same receptor for the same antigen that you're seeing in here. So these T cells can go around and bind to other antigen presenting cells, or can bind to the other MHC molecules which are present at the surface of this macrophage. And keep in mind that these schematic figures are for educational purposes. These are basically conceptual models. So the molecules that I have in here, the molecules that I have in here, like the MHC molecule, they are very small. They are very small, but the cells compared to those molecules are very big. So it's not like a single MHC molecule with a single peptide, with a single co-stimulatory molecule that are involved in activating T cells. No, you have to have hundreds of MHCs with hundreds of peptides, with hundreds of B7 molecules to fully activate a T cell. So, and they're all spread all around it. So the surface of this macrophage or dendritic cell is covered by MHC antigen complexes, as well as the co-stimulatory molecules. So the newly arrived T cells can go and bind to other regions of the same antigen presenting cell. What happens next determines the fate of the T cell. So CD4 T cells are also called T helper cells because they help other cells. But how they help is important. They can provide help in different formats. So if the macrophage in here has phagocytosed something like a mycobacterium, a hard to kill pathogen, it will start producing a cytokine named interleukin 12. And interleukin 12 would tell the T cell to differentiate into a subtype which is called T helper 1. And T helper 1s are able to produce a cytokine named interferon gamma. Interferon gamma goes back and influences the antigen presenting cell. In addition to that, some molecules would appear at the surface of T helper 1 cells that I'm showing you one of them. It's called CD40 ligand, and it binds to the receptor at the surface of antigen presenting cell, which is called CD40. And this leads to some changes inside this macrophage. Well, what is the problem in the beginning? Well, it had phagocytosis, something which was hard to kill by phagolysosome formation. Now T cells have arrived, and under the influence of interleukin 12, they have differentiated to T helper 1, and they are producing interferon gamma as well as this molecule. So it changes stuff inside the macrophage and enhance its microbe killing activity. It's called microbicidal activity. How does this happen? Well, under the influence of interferon gamma, macrophage starts producing a very, very toxic molecule named superoxide. What is a superoxide? It's an oxygen molecule that has an extra electron in it, and it's very, very toxic, and also produces other toxic stuff. Superoxide leads to generation of hydrogen peroxide and hypochlorite, and together these are called ROS, or reactive oxygen species. Reactive oxygen species are very toxic for microbes, and the mycobacterium that was hard to kill can be killed now by the ROS. These mechanism, mechanism is called oxygen dependent killing, to differentiate it from previous mechanisms. And this is a very important process in defense against things like mycobacteria. So tomorrow in the immunodeficiency class, we will see that some people have difficulties in producing ROS. They cannot produce ROS at sufficient levels, and they get a disease. And the disease makes them susceptible to infections like mycobacterial infections. So this is very important. Well, people who had discovered this whole process found out that if you don't have T cells, another cell type can sort of simulate or replicate part of the T cell function. So we have a cell named natural killer cell. And NK cells are also able to produce interferon gamma if they see interleukin 12. So it's like this, like NK cells are always floating around different, like in the blood and sometimes in tissues. When they see a macrophage or an antigen presenting cell, which is producing IL-12, they say that, oh yeah, you're producing IL-12. So I don't know what's happened inside you because I don't have receptors for MHC and antigen. I'm not part of the adaptive immune system. I'm part of the innate immune system. So I cannot recognize antigens. Nonetheless, if I see interleukin 12, I will start producing interferon gamma. And the interferon gamma from natural killer cells can also help by increasing microbicidal activity. So actually, in the real life scenario, when something is phagocytosed, which is hard to kill, the antigen presenting cell starts producing interleukin 12. So the help from NK cells arrives earlier, and the help from T helper 1 cells arrives later. These guys are fast. These guys are slow. NK cells are fast. So in a matter of hours or maybe one day, NK cells are there producing interferon gamma. But the help that they provide is not very efficient. It arrives fast, but it's not very efficient. The help from T cells arrives later, but it's very efficient. So the NK cells are fast, but the help is like not very strong. T cells arrive later, but the help is much stronger. So why the help from T cells arrives later? Well, because T cells, when they recognize MHC and antigen, they need time to proliferate. After that, they need time to differentiate to T helper 1, and then they start producing interferon gamma. But for NK cells, that's not the story. They don't need time to proliferate or differentiate, so they give interferon gamma fast. Nonetheless, help from T cells is much stronger for the same reason, because you have a lot of T cells in here, so they produce a lot more interferon gamma. And in addition to interferon gamma, they have these molecules at their surface, which also help with the activation of macrophage. So these two things should be kept in mind. NK cells fast, but not very strong in terms of helping the antigen presenting cell or the macrophage. T cells, their help needs a couple of days. Actually, if you recall from previous class, when I was talking about type 4 hypersensitivity, I alluded to this concept. I said that type 4 hypersensitivity, which is mediated by T cells, it's also called delayed type hypersensitivity because it takes time. Well, in the context of hypersensitivity, that is the unwanted damaging response. Here we're talking about the wanted useful response. Nonetheless, in terms of the time that it takes, T cells takes 2 to 3 days at least to mount their response. All right, so I have three mechanisms in here to make it a bit colorful. Number one, number two, number three. Number one is the formation of phagolysosome. This is an innate defense. The second step is natural killer cells helping macrophages. So I'm assuming that this antigen presenting cell here is a macrophage. I have made a phrase for it, NK mediated macrophage activation. You can call it NK cells help macrophages. This is another innate defense. And finally, we have T helper 1 cells helping macrophages. I named it T helper 1 mediated macrophage activation. And this is a combination of innate and adaptive defenses. So three mechanisms. So before moving to the next part, I want to ask a question. What if I have phagocytosed something that is easily killed by phagolysosome formation? What if the pathogen that I have phagocytosed is not a hard to kill pathogen? It's an easy to kill pathogen. So mere formation of phagolysosome kills the pathogen. In that case, would it still be necessary for the antigens to be presented to T cells? Well, it seems that, well, it's not necessary, because if the first mechanism is working, why should I bother presenting antigen to T lymphocytes and getting them activated and getting into produce interferon gamma? It seems unnecessary. Well, it is unnecessary for the antigen presenting cell because it can kill the pathogen, but the help from T cells might still be needed by other cells, like B lymphocytes, for instance, because, well, these T helper cells do not only help the phagocytes, they might help B lymphocytes, they might help CD8 T cells, so their help might still be needed. So for that reason, even if the pathogen is an easy to kill pathogen, the antigens would still be presented. The T cells would still come, and some of them would be turning into T helper 1, and interferon gamma would still be produced. But the phagocyte will not need interferon gamma. It will not listen to interferon gamma. How does it listen to interferon gamma? Well, whenever a cell wants to listen to something, whenever a cell wants to get influenced by something, it needs to produce receptors for it. And if you don't have the receptors for it, it means that the cytokine will not influence it. So you won't have reactive oxygen species, but interferon gamma would be used somewhere else. All right. So let's move on to the second space, which is intracytoplasmic space. I said that, okay, the slides are gone. Let me check back. Okay, so for a moment, the slides were gone. Well, they should be back now. Let me check. Let me reshare the slides. Yes, it will be presented. They will be presented anyways. So let me share the screen again. There was something wrong. Okay. Do you see the slides now? All right. I think it should be back there. Excellent. Thank you. Okay. All right. So let's move on to the second space, which is inside cytoplasm. Inside cytoplasm. So the most important group of pathogens which live inside the cytoplasm are viruses. We have things like rickettsia and chlamydia, which live inside cells as well. But viruses are the major group of intracytoplasmic pathogens. So imagine that I have three cells in here, let's say three hepatocytes, liver cells, and the one in the middle is infected by a virus, by a hepatitis virus. So what happens in the first step of defense against intracytoplasmic pathogens is the production of interferons. So we talked about interferon gamma in previous slides. So interferon gamma is an interferon which is only produced by leukocytes, but the ones that we are showing in here are different types of interferons, interferon alpha and interferon beta. So interferon alpha and interferon beta can be produced by a lot of cells in the body. They can be produced by fibroblasts, by epithelial cells, by hepatocytes. And the way that they work is that when they are produced, they influence nearby cells in a paracrine fashion. And they influence the same cell that has produced them in an autocrine fashion. You know, when something is produced by a cell, it can influence the same cell. It's called autocrine fashion. It can influence nearby cells. It's called paracrine fashion. And it can influence cells which are distant from the producing cell. It's called endocrine fashion. So interferons influence nearby cells in a paracrine fashion and the same cell in an autocrine fashion. And they change the internal state of the cell, generating something which some people have called it antiviral state, a state which makes it difficult for viruses to proliferate. So what is this antiviral state? Well, it has different aspects. One major aspect of antiviral state is reduction in the synthesis of proteins. In other words, if you want to sort of anthropomorphize this, like make them similar to human beings, the cell in the middle will tell nearby cells that, well, beware, I'm infected by a virus. So they say, okay, what can we do? Well, it tells them that I'm giving you interferons, and these interferons will turn off protein synthesis inside you. So it tells them basically to turn off their protein synthesis machinery. Don't make proteins anymore. Don't make proteins anymore. So how does it help in protection against viruses? Well, viruses go inside cells to make their own proteins. So if you decrease or turn off the activity of the protein synthesis machinery, it can be a very good antiviral defense mechanism. So that's what interferons do. And of course, here the question remains that, okay, if I turn off the synthesis of proteins, how can I make the proteins which are required for my own survival? So there are answers for that, but it's beyond the scope of this class. So for the interferons, it's sufficient to know that they decrease the activity of ribosomes and tRNAs and other things that are involved in protein synthesis. And that's a first step defense against viruses. It's an innate defense. It's an innate defense. It's not an adaptive defense because we don't have leukocytes in here. Interferon. And interferons have been called interferon. The name comes from interfering. Like if you interfere with something, it means that you're causing problems for that process. So interferons are interfering with the production and proliferation of viruses. The second step of defense is that when you have something inside cytoplasm, its antigens will be loaded at the surface of MHC class 1. So this is different from the previous situation. If it's something inside phagosomes, the antigens will be loaded at the surface of MHC class 2, in the case of phagocytosed antigens. But if it's inside the cytoplasm, they will be loaded at the surface of MHC class 1. And here CD8 T cells would come. And CD8 T cells, they're also called cytotoxic T cells or cytolytic T cells because they kill other cells that are infected with things like viruses. So they release the toxic granules that they have. And they also start showing a molecule which is a deadly molecule named Fas ligand. And together they remove the infected cell, including all of the viruses that are at different stages of development inside the cell. So this is in an adaptive defense. This is an adaptive defense. All right. So killing by CD8 cells. So now we have something which is a very interesting phenomenon. During evolution, viruses and host cells, or let's say microbes and hosts, have always been competing with each other. They've been trying to find ways for pathogens, they've been trying ways to find ways to basically increase their chance of survival. And for host cells, they've been trying to develop ways to protect themselves against pathogens. And this way of killing the infected cell by a CD8 T cell is a type of mechanism that our immune system has developed during millennia, during millions of years, to protect us against intracytoplasmic pathogens. But viruses have also been active in the process. So some viruses have learned that if they block the expression of MHC molecule, if they decrease the expression of the MHC molecule, it will be good for them because this MHC molecule is always trouble. Whenever they infect a cell, the MHC molecule will start showing their antigens to CD8 T cells. CD8 T cells would come and kill the cell which is hosting the virus, and it will decrease the chance of the virus to produce its progeny. So they decrease MHC class 1. And well, one thing to keep in mind is that, as I mentioned, it's not a single MHC molecule. So, like you have to have hundreds of MHC molecules or even thousands of MHC molecules with the peptide for the CD8 T cell to efficiently attack the infected cell. So, if you can decrease the number of MHC molecules, it would be it will be beneficial for the virus. So they decrease the activity, or sorry, the production of MHC molecules, so that CD8 cells cannot kill. But here comes the countermeasure. So some people have called it an arms race. You know, an arms race, like different, like in countries that's in the political arena or military arena, when 2 or 3 different countries are competing with each other, they try to develop weapons and counter weapons. It's called an arms race. And here we have an evolutionary arms race, an arms race in the process of evolution between a host and a microbe. So the microbes decrease our MHC class 1. What can we do? Again, NK cells would appear. NK cells are very interesting in this context. So NK cells do not have receptors for antigens, but they have a receptor which is called an inhibitory receptor. Inhibitory receptors are a peculiar type of receptor. When they see their ligand, they turn off the cell. When they see their ligand, they tell the cell that, okay, you don't need to do anything. The majority of the receptors that we know, they're activating receptors. So when they see the ligand, they will activate the cell. But inhibitory receptors, when they see their ligand, they will tell the expressing cell that, okay, there is nothing wrong, you don't need to do anything. That's why they're called inhibitory receptors. What is the ligand for the inhibitory receptor? The MHC molecule, the MHC molecule itself, regardless of whether it has an antigen or not, because antigens, NK cells cannot recognize antigens. So NK cells usually check the surface of other cells for the presence of sufficient levels of MHC. If MHC is present at sufficient levels, they will be inhibited. They don't do anything. But if MHC is decreased, then they get suspicious. Okay, well, there's probably something wrong with this cell. Let's go and bind to it to see if we can find anything more. So NK cells have a motto. Whichever cell that doesn't produce sufficient levels of MHC should be killed. That's their motto. But before they perform the execution, they check for something else, because we have cells that are innocent cells, normal cells that have low levels of MHCs. So NK cells would say, okay, yeah, maybe you're still an okay cell. I don't know that. Let me double check to be sure that I'm detecting a really infected cell. How do they do that? Well, we have a group of antigens inside our cells. These are self-antigens. These belong to our cells. And these are called stress antigens. The stress antigens are usually inside the cell. But when a cell undergoes a stress, like a physical stress, a chemical stress, or microbial stress, these molecules would move on to the cell surface. And it shows that there is something wrong with the cell. NK cells have a second receptor. It's called NK activation receptor. Unlike the inhibitory receptor, this is an activation receptor. It means that it will get activated, or it activates the cell if it sees its ligand. So they go and bind to stress antigens. And this leads to full activation of NK cells. So there are 2 requirements for NK cell activation. MHC should be low or absent, so that the inhibitory receptors don't see anything. And stress antigens should be present, so that the activating cells activate the cell. And together, when these things happen, then the NK cells would start releasing toxic granules, similar to toxic granules that CD8 T cells had. And through this, they remove the pathogen. So we have different mechanisms in here. By the way, we have a cytokine here again, the interferon gamma is also produced. And the function of interferon gamma in here is to increase the amount of MHC class 1. If MHC class 1 goes up to a sufficient level, then CD8 T cells can perform their killing function. But if interferon gamma doesn't work and MHC doesn't go up as a result of the viral mechanisms, then the NK cells perform their function. And well, it's not part of the class, but we have some clever viruses that have developed ways to even trick NK cells not to kill them. One of them is named the cytomegalovirus. Let me see if I have the slide for it. It's nice to know about it. So we have a virus named, it's not part of the class, but let me show it to you. So we have a virus named cytomegalovirus, CMV. And when it infects the cells, it removes MHC from the cell surface so that CD8 T cells would not kill the cell. But now, well, NK cells would approach and kill the cell. So CMVs are able to make a molecule which looks like MHC, but it's a fake copy of MHC. It doesn't do any work. How can they produce this fake copy of MHC? Well, we don't know, but it's very likely that during this arms race, they have stolen the genetic information for making an MHC molecule from our own cells. In the arms race, we have something named industrial espionage or information espionage. So they send rival countries like Soviets, for instance, during Cold Wars, they were sending spies to United States to steal the information about how to make weapons and stuff so that they can generate something like it. So it's called industrial espionage or weapons espionage. So it looks like that CMV, during millions of years of evolution, has stolen the genetic information for how to make an MHC molecule from us. Then inside the CMV, it's changed some amino acids so that it's not functioning anymore, but it looks like an MHC. So the way that it does is that it removes our own MHC molecule, the native normal functioning MHC molecule, so that CD8 T cells cannot kill. But then they replace it with a fake MHC copy, whose genetic information is probably stolen from us. Some people call it gene hijacking. Like, an airplane, if somebody steals an airplane, it's called hijacking. This is called gene hijacking or gene stealing. And they put it at the surface of our own cells. NK cells would come and see that, yeah, there is something looking like an MHC there. So they say, yeah, you're okay. I'm not going to kill you. On the other side, CD8 T cells cannot kill because the normal functioning MHC is not there. And the CMV lives there inside the cell happily ever after. So that's one interesting, the so-called evasion mechanism. All right, so let's go back to what we had in here. So for intracytoplasmic pathogens, we have three groups of responses. Okay, the first one, interferon response. It's an innate response, production of interferon alpha and beta. Then killing by CD8 T cells. I call it CD8 mediated lysis or CD8 killing, which is an adaptive response. And then we have NK cell killing or NK cell lysis, which basically usually happens when the MHC levels are low and stress antigens are high. So in order to summarize what I said up to this point, I have made this slide. Well, you have to turn your head like 90 degrees to the right in order to read it. So for intravesicular, let's come back in here. We have phagolysis formation, innate defense. Then we have NK cells helping macrophages, like this M5 is macrophage. Then we have CD4 T cells helping macrophages to do what? To produce reactive oxygen species. Here for intracytoplasmic pathogens, we have interferon response, innate defense, we have CD8 response, killing by CD8, and we have NK cell response, killing by NK cells. All right, so in the next 10 minutes, I'm going to tell you about the extracellular space. And some of the slides that I'm going to show you, you've probably seen before. This is the realm of the antibodies. This is where B cells are active, and they can fight against pathogens in the extracellular space. Either pathogens which are going to live in the extracellular space or pathogens that are going through the extracellular space. Their final destination might be inside the cells, but they are going through the extracellular space. They can be targeted by antibodies. All right. So this is the slide that I showed you before. Different classes of antibodies performing different functions. IgA performing neutralization of targets at the mucosal surface. IgE activating mast cells and eosinophils. IgM activating complement, and IgG performing multiple functions, including opsonization, complement activation, and neutralization in serum. So if you have a B cell, and if you put a pathogen next to it, or pathogen antigens derived from the pathogen, B cell will be activated and it will start producing antibodies. But what kind of antibodies? Mostly IgM. So if B cells are alone, if they don't receive help from anybody else, what they do is to produce IgM antibodies, and that is what they have learned during their development in the bone marrow. They also have IgD at their surface, but B cells usually do not secrete IgD. They only produce IgM. IgM is a good antibody, but a lot of functions that we require from antibodies are not performed by IgM. So in order for other antibodies to be produced, we need help from antigen-presenting cells and T lymphocytes. So here I have an antigen-presenting cell. It has phagocytosed the same pathogen, and it's showing the antigen to T cells. And under some circumstances, it also produces interleukin-12. I don't know why this has gone up a little bit, so let me bring it down. So they produce interleukin-12. Okay, there we go. And the T cell becomes T helper 1. What do T helper 1s do? They produce interferon gamma. So in the previous example, when the phagocyte had phagocytosis, something which is hard to kill, interferon gamma would be beneficial for the induction of, or the production of reactive oxygen species and the killing of the pathogen. But in this case, we're talking about an extracellular pathogen. And the antigen presenting cell can kill it inside it easily by phagolysosome formation. So it doesn't require interferon gamma. But interferon gamma here would be helpful for B cells. So it will influence B cells and tells them that, okay, you don't need to produce IgM anymore. What we need now is IgG. Start producing IgG because IgG, especially IgG1 and 3, are more relevant for defense against what I'm going to defend against. Sometimes the antigen presenting cell phagocytoses something, but then it looks at it. Well, this is a very experienced cell. It looks at it and it finds out that these are parts of the worm or a parasite, mostly worms. So, well, it thinks about it and says, yeah, well, worms, if you want to kill worms, IgM is not going to work, IgG is not going to work. I need to tell B cells to produce IgE. But I do not communicate with B cells directly, like there is a hierarchy, okay? So I talk to T lymphocytes and tell them to become T helper 2. How does it do that? Well, if you present an antigen to T cells, but do not give them interleukin-12, the T lymphocytes will gradually start differentiating themselves towards something which is called T helper 2. And T helper 2 produces interleukin-4, which accentuates this process. It increases the production of T helper 2 cells until they produce a lot of interleukin-4. And interleukin-4 would tell B cells to produce IgE, because IgE is more relevant in here. Well, what else do we have? In some cases, the antigen presenting cell, if it's located underneath mucosa, like gastrointestinal mucosa, underneath mucosa, it phagocytoses the thing and then it looks at it. Well, it has mechanisms to sort of analyze what it has phagocytosed. And it figures out that this is coming from a pathogen which is inside the gastrointestinal lumen, like it's on the other side of the mucosa. So it thinks that, okay, well, IgM cannot be secreted through the epithelial layer to the mucosa. So it's irrelevant. IgG cannot be secreted, and IgE cannot be secreted either. So I'm going to tell the T cell to differentiate into something that some people have called it Th17, sorry, T helper 3. And T helper 3 would tell B cells that they need to start producing IgA. IgA is the antibody which can be secreted. So IgG, it's used when the pathogen is inside serum and it is to be neutralized, like it could be a bacterium inside serum or a bacterial toxin, or more importantly, viruses which are inside body fluids. They have not got internalized by cells yet. When we want to opsonize something or when we want to activate complement. Well, complement could also be activated by IgM, but these two other functions are IgG specific. How about IgA? Well, it helps in mucosal defense when you want to secrete antibodies to mucosa so that it protects inside the gastrointestinal lumen. And how about IgE? Well, I showed you IgE in previous class. When you have a worm, you want to kill it, you cover the worm with IgE so that eosinophils and mast cells would come, they would bind to the surface of the worm. And again, keep in mind that these pictures are not drawn to scale. Worm is very big. These cells are very, very tiny and small. So it's not like a couple of eosinophils and mast cells binding to the worm. Like thousands of eosinophils will cover the worm. Thousands of mast cells would cover the worm. It's not a single, uh, basically, cell. There are thousands of them. And then the eosinophils will start releasing their toxic granules, and mast cells would do the same, and it kills the helmet. It kills the helmet. Keeping in mind that, as I mentioned that in the previous class, if it's a worm which is inside tissues, the role of the eosinophils is more important. If it's a worm that is inside the gastrointestinal lumen, the role of the mast cells would be more important. Histamine and other mediators from mast cells would make smooth muscles in the gastrointestinal tract to contract so that the worm is dislodged from its location. They will increase the production of mucosa. That's another way to dislodge the worm. And sort of the responsibilities are divided between eosinophils and mast cells. All right. So this shows us the picture for defense against pathogens outside of the cells. And I have one more cell to add in here. Like almost 15 years ago, another group of T cells were discovered. And these T cells were a peculiar type of T cell that required several cytokines to be differentiated. And they were called T helper 17. T helper 17. So why on the word 17 all the way from 1 to 3? Well, they should have been called T helper 4. Why 17? Because there was a cytokine at the time that was an orphan cytokine, ORPHAN, orphan cytokine. It was named IL-17. So when we say orphan for human beings, if somebody's lost his parents, it's called an orphan person. But for cytokines, an orphan cytokine is a cytokine that you don't know who is producing it. So you know that it exists. And people knew for many years that there is something named IL-17, like they had named interleukins 1, 2, 3, 4, all the way to 17. So this was named interleukin 17, but nobody knew who produces interleukin 17 until this cell was discovered, and they named it after the cytokine that produces it. It's a neutrophil activator. It's a neutrophil activator. Like in Arabic or in many languages, sometimes children are named after their fathers. Sometimes fathers are named after their children. Like somebody has a son named Hassan, for instance, in Arabic, and the father will be named Abu Hassan, the father of Hassan. So this is like that. This TH17 is basically Abu IL-17, the father of IL-17. Or you can call it IL-17 if you want to consider it mother. So it's Abu IL-17, the father of IL-17. That's why it's got the name 17 in it. And IL-17, it's one of its sons, one of the cytokines that it produces, and they activate neutrophils. Especially when you have a lot of neutrophils, you need a lot of neutrophils. IL-17 would be very useful. And this Abu IL-17 has also another son or daughter, and it's named interleukin 22, but it's not been named Abu IL-22 because, well, the 17 is first. So interleukin 22 will influence epithelial cells, and tells epithelial cells that you can also be involved in defense against microbes. How they can do that? Well, epithelial cells form a physical barrier. That's the first thing. But they can also produce peptides that are named antimicrobial peptides. And these antimicrobial peptides can be involved in defense. So that's how the story works. All right, so this is almost completes the story for defense. I'm going to use a metaphor in here just to help you memorize this whole thing. In armies, in classical armies, you have a hierarchy. Like a hierarchy is like there are different levels of authority. So you have a commander, the army commander, central army commander at the top. Central commander is basically a veteran soldier, a soldier who's been fighting a lot for decades, but now he's very good in devising military strategies and giving orders to other people what to do. So that's the top commander. Then you have high level officers or top level officers that are in contact with the central commander. Then you have lower ranking officers that get their orders from high ranking officers. And then you have the regular soldiers, ordinary conscripts and soldiers. And in regular armies, like you have a central commander, maybe then you have 10, 20 top officers, and you have maybe a few hundred lower ranking officers, and then you might have hundreds of thousands of soldiers. And you also have ordinary people, especially people who live in the border areas, because that's where invasion happens. So if I want to look at this picture in here, in terms of a hierarchical structure, who is the central commander, you think? Can you type anything? Who is the central commander in here, in this structure, that in this simplified representation that we have in here? Who gives the orders to other people, to the top level officers, what to do? Let's see if you're still with me. All right. Very good. Okay. Excellent. The antigen presenting cell is the central commander, which could be a macrophage or a dendritic cell. This is an old veteran. He can fight very well. Well, for dendritic cells, they are usually not involved in fighting. They mostly give orders, but macrophages fight at the same time. But they are also very good in devising military strategies, telling other people what to do. Who are the high ranking or top level officers? CD4 T cells are the high ranking officers, and they get their orders directly from the central commander. Sometimes it tells them to become T helper 1, sometimes it tells them to become T helper 2, or maybe T helper 3 or 17. We have another T cell also called T regulatory that I have not included in this picture. It has regulatory functions. So these are top level officers. How about the lower ranking officers? Well, B lymphocytes are in this hierarchical structure that I'm drawing are kind of low ranking officers. And also CD8 T cells. They get their orders from T helper cells or CD4 T cells. So how about ordinary soldiers? Well, neutrophils are the ordinary soldiers. We have a lot of them. They fight very well, but they die very fast also. So they fight very well. If they leave them alone, they fight very well. But if they have a commanding officer organizing them and giving them motivation and morale, they would fight much better. So this TH17 or ABOIL17 is the officer that is commanding the neutrophils, the ordinary soldiers. The rest of them are mostly commanding B cells and CD8 T cells. How about the ordinary people? Well, the ordinary people, especially the ones that are located at the borders, are epithelial cells at the border regions. So they provide a defense of the physical type first. But then, if they receive some motivation in terms of IL-22 from Th17 cells, they would mount a stronger defense by producing antimicrobial peptides. So this is how the entire set of molecules and cells work. And here is the summary slide. So here I have if something is inside the blood or interstitial tissue or lymph, T helper 1, interferon gamma, B cell, and IgG. If it's a parasite, T helper 2, interleukin 4, B cell, and IgE. If it's at the epithelial surface, well, some other T cell, it's controversial whether to call it T helper 3 or not. So some other type of CD4 T cell. Then a cytokine named TGF beta, B cell, and IgA. And then we have two spaces for T helper 17s. If it's somewhere, the pathogen is somewhere that neutrophils can have access to, then TLPR17 producing interleukin 17 and activating neutrophils. And if it's at the epithelial surface, TLPR17 producing interleukin 12, telling epithelial cells to produce antimicrobial peptides. So this is the entire structure. And it's a very colorful slide. It summarizes almost everything that I said in this class. And yeah, it's the end of the class. I have a quiz for you in here. And then we have some more slides that is just for the reading. They are about evasion mechanisms. Evasion means basically uh, uh, escaping from immune response that I'm not going to teach you. I just told you about one example. That was the CMV example. So that might be included in the exam, the CMV example. But the rest of it, I don't expect you to read it for the exam. Just read it for your own interest. So, okay. So here is the quiz. You can answer the question A, B, C, D. The question is this: which of the following is not generally a mechanism of response against viruses when they are inside cytoplasm? What is the answer to this question? Which of the following mechanisms is not considered a mechanism of immune response against viruses? A, interferon response, lysis by NK cells, lysis by CD8s, and activation of macrophages by NK cells. Let's see who can answer. And all of you should answer, actually. That's just to be sure that you're still with me at the end of the class. Okay, let me come back here. Thank you. Thank you very much. Very good. So D is the answer. Right. D is the answer. So the question is asking which one is not considered the mechanism of defense. And if I come back to here, for intracytoplasmic, I'm talking viruses, intracytoplasmic. So it means here. So interferon response is one mechanism, like interferon alpha and beta. Killing by CD8 is another mechanism. Killing by NK is another mechanism. But activation of macrophages by NK cells is usually not a mechanism for defense against intracytoplasmic pathogens. It's in here. It's the mechanism of defense against intravesicular ones. So the correct answer in here would be answer D, macrophage activation by natural killer cells. Excellent, very good. End of the class. Thank you. Thank you for your presence in the class. And all right, so I'll see you tomorrow at 8 in the same place. And we will talk about, thank you very much. We will talk about immunodeficiency. We will have like a part of the class, maybe 70-80% of the class will be talking about primary immunodeficiency, genetic disorders that lead to insufficient immune response. And then we will also talk briefly about, thank you all, about HIV and AIDS. So if you wanted to see this, the movie for the class, you can come back to this channel. It's my YouTube channel, NoorBioScience. And then you go there and you click on live. If I can show it in here. So you click on live and so these are the videos. Well, they are unfortunately, most of them are in Farsi. I have some English classes too. I'll add some more. But if you come back to live, you will see them. This is the live session that we're doing now. You see the live in here. But the previous classes in here, mechanism and classification of hypersensitivity. And then the immunology class, infectious disease, is going to be here. All right. So thank you very much. Thank you all, and I'll see you tomorrow. Thank you. Take care.